Hello, everybody. My name is Stefan Möser. I'm the CEO of NovaGo Therapeutics, and I'd like to tell you a little bit about how we want to develop antibody therapeutics to unlock nerve regeneration in acute spinal cord injury. We are a small biotech company founded out of the University of Zurich originally, have two assets we're working on, one in CNS, which I want to talk to you about, and a second one in ophthalmology, which we'll probably leave for another time. For both of them, we think that this could be a first-in-class therapy, and all the IP belongs to us. We're a very small management team of 3 people. You can see it here with a scientific founder, Professor Martin Schwab, a CEO, Roger Vetli, and 5 additional employees in our clinical team. I don't have to introduce you to spinal cord injury, but what I want to make sure is to really highlight that what we're doing here is to regenerate the neurons which have been degenerating after the injury. So it's neurite growth and new connections which you want to form. The biology behind is around the inhibitory factor NoGo-A, which is a major inhibitor in nerve growth. And NoGo-A is a transmembrane protein on neurons and oligodendrocytes. And we've developed an antibody, NG004, which binds to the extracellular loop of NoGo-A, and by this prevents the interaction with this receptor. It's one of the stopping factors, so as long as NoGo-A is present on the surface of neurons, neurite outgrowth is inhibited. Professor Martin Schwab was the one who discovered this protein quite some time ago, and we developed several antibodies against this over the years. And let me explain you a little bit like the timeline, how NoGo-A was discovered and how the different events came together. Between 1988 and 2000, Martin and his lab discovered and described NoGo-A the first time, and a lot of the initial mechanisms were analyzed then up to 2005. In 2005, the first clinical trials started, and Novartis, together with Martin, started a phase 1 safety study. This was followed, unfortunately, by strategic deprioritization of Novartis because they did close their disease area for neurology at that time. But Martin was able to convince them to donate the antibody to an academic consortium called the NISCI Consortium to run an IIT study. This NISCI Consortium, which was able to run an IIT phase 2 study, was able to give this antibody to patients between 2019 and 2023, and I will come to the results in a second. NovaGo Therapeutics was founded in 2015 because it became clear early on already that there's a new antibody required to push this further, and that's when the discovery of new anti-NoGo antibodies started. The NISCI Consortium was able to do an IIT trial with 129 tetraplegic patients and do this in 13 European clinical sites. And you can see it was founded by the European Union, by the Swiss Paraplegic Foundation, by Wings for Life, and was quite an interesting clinical consortium. They were able to go through this study in several subpopulations, and there is a nice publication which came out in Lancet Neurology last year, and I will leave the details to this publication. The 2 things, though, I would like to highlight for today. On the upper part, you can see the SCIM self-care score for motor incomplete patients. So it's a subgroup of the acute spinal cord injury population. And there we can clearly see that 6 months after the anti-NoGo treatment, there's a big difference between the placebo group and the antibody-treated group. On the bottom, you can see that there's also a nice effect on the walking ability of these patients. So you can see here for the SCIM mobility, there's an item 8, and the item 8 describes the walking ability without walking aids. And you can see that there is a huge difference between placebo and the drug-treated patient population after 6 months. So the mobility of these patients changed dramatically. And as I said before, please have a look into the paper. The reference here is on the right side. Happy to discuss more about that in separate meetings as well. What we did then is we used the Neurimmune human antibody pipeline to find a new antibody against NoGo-A. So this pipeline utilizes the immune repertoire of healthy elderly donors, which then are mined for specific antibodies against certain targets, and then put into validation and further characterization. NG004 came out of this pipeline, was modified to be an IgG4 with limited or no effector function, and then used for further testing. The difference to the old Novartis antibody in this case is that it's an IgG4 which has the S2 to 8p mutation, which is nowadays required by the regulators to prevent Fab-arm exchange. We have a long patent life, and we're aiming for a high clinical dose because we have a good solubility and the biophysical properties of the antibody are excellent. The antibody can be manufactured in standard show cell systems, which also will enable a much easier transition to a larger scale production at a later time point. In vitro, this is one experiment of many, which shows how the antibody can work. So these are cells which are treated with monkey spinal cord extract. And as you can see in the middle here, if you add the antibody, these neurites will start to outgrow. If you just add a control antibody, the neural outgrowth is inhibited. And this assay can be used to be quantified, and we can define EC50s out of this and understand the activity in vitro of the antibody in detail. One of the many in vivo experiments which were done, you can see here at the bottom, it's done in a rat model where the hind limbs of the rats are paralyzed by physical injury of the spinal cord. And then when you look at the curve in the bottom left, you can see a locomotion index where you can see after the injury, animals treated with a control antibody here in blue are recovering, but they're not recovering to the full score again. But you can see if you treat them with an anti-NovaGo antibody here, there's a huge difference, and the animals nearly recover to 100% function again in this locomotion score. This resulted in that we said, okay, well, we really should bring this antibody into the clinic and prepared the full preclinical package. So the antibody went into full CMC production. GLP tox was done in a huge monkey study, which came out clean. And this resulted in start of a clinical trial in August 2024. And the actual first patient was administered to the trial in December last year. This trial looks as follows. It's a phase 1b study in acute spinal cord injury patients. The official primary readout is safety, but of course we're looking at all the endpoints for efficacy as well because we want to see early signs of efficacy in this trial as well. As this is a safety trial, it's a multiple ascending dose study. We have 4 dose groups at 50 milligrams, 100 milligrams, 200 milligrams, and 400 milligrams. And each of these will go through 6 dosings every 5 days in the first 4 to 28 days after injury. Currently, we're at the stage in this trial that we're halfway through. Once the trial would be finished, latest by Q3 next year, most likely earlier because the recruitment so far is going pretty well, we would follow up with a phase 2 study or a 1b efficacy study, aiming for market authorization in 2033. The study itself looks like follows. It has 4 cohorts from 50 milligram to 400 milligram drug injection per injection. There are 6 injections, as I said before, 5 times apart. The first 2 cohorts are finished already, and we just started recruitment for the 3rd cohort at 200 milligrams. Just as a reminder, the antibody used before in the NISCI trial was administered at 45 milligrams only, and we believe from preclinical data out of animal studies that the half-life of the antibody in the spinal cord is relatively short. So a dose of 45 milligram most likely would have resulted in an underdosing of the patient. That's why we want to increase the dose dramatically here, which was not possible with the old antibody, but with this antibody it's possible to overcome a longer dosing holiday. Our PK calculations indicate that all the doses above 100 milligram already should result in substantial drug exposure, which would be good enough to have an inhibition over the entire period. We expect that the last patients will be dosed by the end of the year so that the study could conclude in Q1, latest Q2 next year, but this of course depends on the recruitment rates. The study itself is a multinational study with 2 centers in Switzerland and 4 centers in Germany. And so far, the enrollment rates are actually pretty nice. On the financial side, we are fully financed for the spinal cord injury trial, as I described before, until the end of next year. And we're currently starting our race to finance the proof of concept studies. We have a second asset, as I indicated in the beginning in the introduction, which I didn't discuss today. For this, we're also progressing further, most likely as part of a subsidiary, and are raising money there also to go to proof concept studies. In total, for spinal cord injury, for the race, we need about 25 million to go up to proof concept stage. And for the ophthalmology asset, depending until which stage, it's between 25 and 40 million as well. Thank you so much for your attention, and looking forward to interesting discussions as part of this meeting. Thank you.